All right, so here we go. We're going to start off by talking about an experiment that comes up in several units in the course. It's called the Miller-Urey experiment. It was done in 1953. It's been repeated many, many times. But essentially what Miller and Urey wanted to show was that inorganic substances, substances that didn't come from living things, that weren't really carbon-based, could be used to make organic substances. So they took gases that they thought were in the Earth's early atmosphere, things like water vapor, hydrogen gas, H2, ammonia gas, NH3, and methane, CH4. They put those in a chamber like the one you see down here at the bottom of the screen. In a part of the chamber, they put some water, and they heated that to boil it. They connected that up with a tube so that the water vapor went into the chamber with the other gases. And in here, there was a spark plug kind of setup so that every so often those spark plugs made a spark. That was supposed to simulate lightning. Eventually, the gases cycled back through. And what they found after several days is in this collection container, they found organic molecules, things like amino acids, things like nucleotides, the building blocks of DNA and RNA. So what they were trying to show is that from inorganic precursors, organic molecules could have been made naturally through things like the addition of energy, whether it was from lightning or from heat, could have been like volcanic heat, and just random reactions between those chemicals. So Miller and Urey showed that organic molecules could be made from inorganic precursors. And they did that in 1953. Really important experiment. All right, the next thing we're going to talk about is water. Water is obviously hugely important in biology. You guys know that water is H2O, and that the bond that holds the H and the O's together in a single molecule, this bond or this bond, that's a covalent bond. That's formed by the sharing of electron pairs between oxygens and hydrogens. That bond is very strong. It's hard to break. It's an intramolecular bond, meaning that it's inside the molecule. Now, when they share electrons, they don't share them equally. Oxygen is a little more, is actually quite a bit more electronegative than hydrogen is, which means when they share electrons, oxygen pulls those electrons closer to it and further away from the hydrogen. So it's not equal sharing. And what that causes is that it causes more negatives to be concentrated near the oxygen and less negatives to be concentrated near the hydrogen. And that causes the oxygen to have a slightly negative charge and the hydrogens to each have a slightly positive charge. This is not like +1 or -1. It's just slightly positive, slightly negative. And because of the shape of the water molecule, which is said to be bent, with the hydrogens on one side and the oxygen on the other, the water molecule is said to be polar. It's polar because it has one side that is distinctly negative and another side of that molecule that's distinctly positive. So the oxygen side is negative, the hydrogen side is positive. And what that causes to happen is that it causes water molecules to stick to each other, because this negative oxygen is going to be attracted to positive hydrogens on another water molecule. We can see that happening right here. That attraction is called a hydrogen bond. Bond is probably not the best word for it because it's not an intramolecular bond. We want it to refer to those bonds, like hydrogen bonds, as intermolecular forces. And I'm hoping that you know, and if you don't know, you need to know before this day is over, inter is a really common prefix in AP Bio, and it means between. So hydrogen bonds are forces of attraction between water molecules. not within a water molecule. They're between different water molecules. And you can see that happening here, you can see it happening here, and you can see it happening here. Compared to covalent bonds, hydrogen bonds are very, very, very weak. Now, you might have heard your chemistry teacher say that they're strong. They are strong compared to other intermolecular forces. But compared to covalent and ionic bonds, intramolecular bonds, they're very weak. But in a container of water, there are millions of them. So even though individually those bonds are pretty weak, collectively, they're very, very, very strong. And they're responsible for most of the biologically important properties of water. Let's talk about what some of those are. So several properties of water that are really, really important in AP Bio. And guys, I hear a mic on. If you guys would make sure you mute your mics, that way we won't get all the background noise as we're going through this. Let's see if I can mute that. There we go. So one of the important properties of water we want to talk about is that water has what we call a very high specific heat. Specific heat refers to the amount of heat needed to change the temperature of 1 gram of a substance by 1 degree. Since water's specific heat is high, that means it's really hard to raise or, on the flip side, to lower the temperature of water. It's really good at moderating temperatures. Water has a high specific heat because of those hydrogen bonds. Remember that heat is really a measure, or temperature rather, is a measure of how fast water molecules are moving. Before the speed of water molecules can be sped up, the bonds between those water molecules are broken. So it takes energy to break those hydrogen bonds before the water molecules can speed up very much. So due to water's very high specific heat, it's really good at helping organisms to maintain a constant internal body temperature. Most living things are made mostly out of water. And one of the important reasons for that is that water acts as a natural coolant or a natural antifreeze. It helps us to maintain a constant temperature. This is water's high specific heat is also important for environments that are near large bodies of water, coastal areas. They have a much more moderate temperature than places that are away from bodies of water, because the water helps to moderate both the air temperature and the temperature of the water itself. So water helps to create a constant internal environment, and it also helps to create a more stable external environment for organisms. Water is also a really good solvent, which means it's really good at dissolving things. Sometimes water is called the universal solvent. It's best at dissolving salts or molecules that are polar or ionic. Ionic are things like salts, positive ions and negative ions connected together. But water is also good at dissolving other things that are polar, like alcohol, for example. This is really, really important in biology because the nutrients and the gases that we need to transport through our bodies have to be able to be transported through our blood. And if they won't dissolve in our blood, it's really hard for our body to transport and distribute those resources. Water also has a really high heat of vaporization. To vaporize it, again, those hydrogen bonds have to be broken. Heat of vaporization is essentially the amount of heat that's needed to convert a liquid to a gas. It takes a lot of heat absorption to do that for water. And this is the main reason that sweat works. Liquid water absorbs lots and lots of heat before it evaporates, turns into a gas, and then takes that heat away from the body with it. Makes sweat work. Transpiration in plants also helps to cool off plants. Transpiration is essentially the movement of water up through the plant, and then it evaporates and leaves the leaf, taking with it some heat from the plant. So both sweating and transpiration are important ways to lower temperature, and they only work because water has a high heat of vaporization. Water is also cohesive and adhesive. Cohesive means water sticks to itself. And we know that happens because water's polar and that water molecules form hydrogen bonds between each other. Water is also adhesive. That means it sticks to other polar things. For example, the xylem, the water tubes in a plant, are polar, and that helps water to move through it. The roots are polar, and that helps water move into the roots of a plant. Another important property is that water expands as it freezes. This is really important for organisms that live in aquatic environments, say small ponds or lakes. As the water gets cold and starts to freeze, the ice expands because the hydrogen bonds that connect chains of water molecules push them further apart. This decreases the density of ice, and it makes ice less dense than water. And because it's less dense, that means ice floats on top of a body of water. Once a layer of ice floats up top, that works as insulation, and it helps to moderate the temperature underneath. If ice sank, if it became more dense as it floats, or as it cools off, like most things do, eventually the whole lake would freeze. Ice floating on top insulates the water underneath, keeps that body of water from freezing solid. You're going to be hard pressed to find a body of water that's completely frozen solid. If it is, it's going to be a pretty shallow, small body of water. So these are all hugely important properties of water. They're really important for life, and most all of them happen because of the polarity of the water molecule. All right, the rest of our session today is going to be devoted to organic chemistry. So this is about the chemistry of carbon-containing compounds. Most of your body's mass is made out of water, but about 30% of the organism's dry weight is composed of carbon. And this carbon is found in organic molecules. Molecules made of carbons connected to other carbons, and usually also connected to hydrogens. We're going to talk about 4 groups of what we call organic macromolecules. They are hugely important in almost all living things. And those 4 groups are carbohydrates, which we'll also talk about as sugars, lipids, which we'll talk about as fats, oils, and waxes, proteins, which we typically just call proteins, and then we've got nucleic acids, which include things like DNA, RNA, and ATP. So those 4 groups are the major forms of organic macromolecules found in your body. The original source for all the carbon in essentially all forms of life is carbon dioxide. Carbon dioxide is not considered organic because it doesn't have carbons connected to carbons or carbons connected to hydrogens. CO2, for example, is the source of carbon for photosynthesis, which ends up being the source of carbon for essentially all other life. So let's talk a little bit about the carbon atom and why it's the atom that most life is built out of. Carbon is in group 4A of the periodic table. That means it has 4 valence electrons. And because of that, it can form 4 covalent bonds at any time. This allows it to bond to lots of different things and form molecules with a variety of shapes and functions. Because you guys, I hope, know, this is a huge theme in AP Bio, that structure determines function. So because carbon can form molecules with so many shapes, it also can form molecules with lots and lots of functions. That's only possible because of those 4 bonds that it can form at a time. The carbon atom is also really small. That means its valence electrons are very close to its nucleus. So when it shares those electrons with other things, the bond that forms there is held onto very tightly. Carbon forms really strong covalent bonds. And again, this makes it a really good building material, strong bonds. Carbon's also really abundant on Earth. It's not the most abundant element on Earth, but there's plenty of carbon on Earth. It's a really good element for building life. There's plenty of it. It makes really strong bonds, and it can make molecules in lots of different shapes with lots of different functions. So very small organic molecules are called monomers. Most of the biologically important molecules we'll talk about, organic molecules, are large. We're going to call those macromolecules. We'll also call them polymers. And these macromolecules or polymers are made of usually many of the small individual organic molecules, the monomers, hooked together. Those are typically hooked together by strong covalent bonds, hard to break. And I want to go to another slide to talk about these two types of reactions. So here we have a monomer, and here we have another monomer. In this case, these monomers are going through what's called a dehydration synthesis, or you can also sometimes hear these called condensation reactions. These reactions build polymers. So they put together monomers, almost like sticking together Legos to make a bigger structure. So here we can see this OH on the leftmost molecule and the H on the rightmost molecule. During a dehydration synthesis reaction, those are removed. Well, that opens up a bond site both on this molecule and on this one. So those molecules join together. We can see that bond right here. And the H and the OH come together to form water. And that's why you hear these called dehydration or condensation reactions. Oftentimes, this requires an input of energy to make this bond form. Not always, but usually. So dehydration or condensation reactions take monomers and put them together to make bigger molecules, polymers or macromolecules. And in the meantime, they release water. The opposite of a dehydration or condensation reaction is called a hydrolysis reaction. This term lyse or lysis is a really important root in AP Bio. Lyse means to break or cut or split. So in this case, we have a large molecule, a macromolecule. Water is added in as a reactant. And when that happens, a bond is broken. And we can see the OH from the water ends up attached to one piece of the macromolecule, and the H ends up attached to the other piece. Point being, hydrolysis reactions split or break large macromolecules down into monomers or smaller pieces. Typically, these reactions are going to release some energy when the bonds are broken. All right, so let's talk about our first major group of macromolecules. And we're not going to spend a lot of time on these. Going to try to move through the first two groups very quickly. We're going to spend most of our time today talking about proteins. Carbohydrates are also known as sugars. In living things, carbohydrates are sources of quick energy, and they're also surprisingly used as structural or building materials. Most carbohydrate names end in the suffix OSE. So if you see an OSE in bio, it's usually a sugar. The monomers of carbohydrates are called monosaccharides, and that just means one saccharide essentially means sugar, one sugar. They're the building blocks of essentially all carbohydrates. Glucose, fructose, galactose, ribose, and deoxyribose are the common monosaccharides we'll talk about in AP Bio. And you've heard of most of those probably before. Monosaccharides have the general formula CnH2nOn. For example, glucose, in glucose, there are 6 carbons. Well, this formula then tells us there's going to be twice as many hydrogens. So glucose is C6H12, and our formula tells us there's the same amount of oxygens as there are carbons, so O6. That's an important formula for you to know. C6H12O6. That's glucose. Turns out it's also the formula for fructose and galactose. They have the same formula, but different shapes. And we'll talk more about that later on in the course. Polysaccharides are the big sugars, the big carbohydrates. They're polymers. They're made of lots of monosaccharides connected together. The polysaccharides that we'll talk about in the course are starch, which is used as an energy storage molecule in plants. And then it ends up being a major food source for humans and lots of other animals. Starch is what rice is mostly made out of, potatoes, corn, and lots of other food crops. Glycogen is a form of sugar that animals use to store excess energy. For example, we store glycogen in our muscles and in our liver. Cellulose is a structural material. It's the main building material in plant cell walls. It's made out of glucose. Turns out that all three of these are polymers of glucose. Glucose molecules hooked together in different patterns, different shapes. Cellulose turns out to be the most abundant organic molecule on Earth because it's in plants, and it's in essentially the whole plant. Due to the way the glucose molecules in cellulose hydrogen bond, it is very strong. It's also very hard for most organisms to digest. Cellulose is sometimes in our diet referred to as fiber, because after we eat it, we really can't digest it. It's just used to push things out of our body. Chitin is another polysaccharide that we'll talk a little bit about in this course. It's a component of the exoskeletons of some insects. It's also found in the cell walls of most fungi. So it's a strong building material. Here we can see some carbohydrates. Oftentimes you're going to see them in ring structures. And at every bend in the ring, unless it's noted otherwise, there's implied to be a carbon atom sitting there. So there's carbon sitting at each of these bends. Notice this one has an oxygen there. So this is glucose. Notice this is glucose with a little different conformation, slightly different shape. And that matters because glucoses with different shapes hook together in different ways. Here we see linkages between the glucoses in starch, and here we see linkages between glucoses in cellulose. And those differences end up causing differences in shape, which in turn cause differences in functions. All right, that's all we're going to say about carbs for today. Next, we're going to talk about lipids. So lipids are macromolecules that include fats, oils, waxes, and also steroids like testosterone and estrogen. Most lipids are hydrophobic. They're hydrophobic because they're mostly non-polar molecules. They contain very little oxygen or other electronegative atoms. They're mostly made of carbon and hydrogen. We're going to first talk about, let's talk about a fatty acid first of all. A fatty acid is a long chain of carbons and hydrogens, only carbons and hydrogens. And then there's also an important building block of most lipids called glycerol. This is a 3-carbon alcohol. And in at least some fats, it acts as essentially the base. And fatty acid chains are connected to that glycerol molecule, making what we call a triglyceride, which is the fat we'll talk about most in this course. These are your basic fats or oils. Up here in the picture, each one of these in dark gray, those are fatty acid chains. And then each one of those is connected to this molecule, which was the glycerol molecule. So this is what we call a triglyceride. 3 fatty acids connected to 1 glycerol molecule. And for right now, we're not going to talk about the differences between saturated and unsaturated fats. We'll do that later. I want to talk about another important type of lipid. But before I do, let's go back to triglycerides for a second. Triglycerides are the fats that we tend to eat in our diet. They are very good energy sources. So animals tend to use lipids to store what we call long-term energy. They're also used for cushioning in the body and also insulation. Really important in aquatic mammals like seals, walruses, whales, to keep the body temperature constant. Let's talk a little bit though about phospholipids. These are another really important type of lipid. These are the main components of the cell membrane. So we have a glycerol molecule, and you can see the glycerol molecule right in here. Connected to that glycerol molecule are 2 fatty acid chains, which we see down here on the bottom, and one structure that's called a phosphate group. This phosphate group essentially started out as PO4-3, the phosphate ion. Notice that that's charged. It's very polar. So this end of the lipid becomes very polar, and this end made of carbons and hydrogens is nonpolar. Because it has a polar side and a nonpolar side, we refer to a phospholipid as amphipathic, meaning that it has both. And once we put these in water, the polar sides of this molecule, the phosphate groups, want to be near water. So they stick out toward the outer environment of the cell and also in toward the inner environment of the cell because there's water in both places. The fatty acid tails end up orienting towards each other. So phospholipids, at least in living things, tend to form what we call bilayers. Two layers where the fatty acids can be toward each other and away from water, and the phosphate heads can be toward the water on both sides, both the inside and outside. So these are the major structures in the cell membrane of all life, the phospholipid bilayer. Not only do they make up the cell membrane, they also make up the membranes around most of your organelles, like mitochondria, chloroplast, even things like lysosomes and vacuoles. We'll just mention steroids for today. So these are some common steroids found in the body. Here we see estrogen, sometimes called estradiol. Here's testosterone, progesterone, another female sex hormone. Here's cortisol, that's a stress hormone. Aldosterone and corticosteroids, or corticosteroid, other steroids important in the body. Remember, with those ring structures, everywhere there's a bend, there's implied to be a carbon unless something else is notated. So these are carbon ring structures. For the most part, they're nonpolar, which means that unlike lots of other hormones, these can penetrate the cell membrane and they can bind with the receptors inside the cell, intracellular receptors. This allows them to actually go into the nucleus and help to regulate the expression of certain genes. So oftentimes, steroids like testosterone and estrogen particularly go through the cell membrane, through the nuclear membrane, and then attach to the DNA and help to either turn off or turn on certain genes in the body. Cholesterol, another steroid, is important in the cell membrane. It helps to keep the membrane flexible, and it also helps to keep it from being too liquidy. All right, the most important group of macromolecules we're going to talk about today, though, arguably, are proteins. Proteins are found throughout your body. Proteins are components of bone, muscle, skin, hair, but they also have lots of other functional components. Most of your body's chemical signaling molecules are made out of proteins. Also, most of your body's enzymes, the molecules that actually catalyze your body's chemical reactions, most of them are proteins as well. So these are hugely important. And something else that should clue you into how important proteins are, really the only function of DNA is to code for the body's proteins. DNA tells your body which proteins to make and how to make them, and that's really all it does. Proteins make up more than 50% of an organism's dry weight after you take out the water. So they are critically important. Typically, their names end in LIN, except for enzymes, whose names often end in the prefix or suffix rather, ASE. So if you see a chemical ending in LIN or ASE, that should clue you in. It's probably talking about a protein. Proteins are usually huge macromolecules made out of monomers, building blocks, called amino acids. We're going to look at a picture of an amino acid on the next slide. Turns out there are 20 different types of amino acids that make up the proteins in all living things. So if you thought of yourself as some Lego creation, only 20 different types of Legos make up all of life. So here we see an amino acid. You should know the parts of an amino acid. So in the middle is a single carbon atom. Connected to one side of that is a single hydrogen atom. Connected to another side is a group, NH2. It's called an amino group. And then connected on the opposite side of the amino group is a group called the carboxyl group. The fourth, and these components up here that I have circled right now are the same in all 20 amino acids. The part that makes the 20 different is what's connected here. Call that the R group. And we're going to talk more about what those R groups might be here in just a second. The amino group can act as a base, meaning that it can accept hydrogens. The carboxyl group, on the other hand, can act as an acid. It can give off hydrogens. So amino acids are sometimes referred to as amphoteric. And what that means is that they can act as both an acid and a base, essentially at the same time. They can release hydrogen ions like acids, or they can accept hydrogen ions like bases. So to make a protein, individual amino acids, the monomers, are connected together. This happens during a dehydration synthesis reaction. And we see that happening here up top. When that happens, the OH from a carboxyl group is removed, and one of the hydrogens from an amino group is removed. So we end up getting a bond between the carbon in the carboxyl group on one amino acid and the nitrogen in the amino group on another amino acid. That bond is a covalent bond, and it's referred to as a peptide bond. When we hear that word peptide, you need to think about protein. It's very strong. It's very, very hard to break. And again, the reaction that put those amino acids together is a dehydration synthesis or a condensation reaction. Two amino acids connected together are called a dipeptide. Typically, proteins, though, are made of several chains of amino acids. More than two amino acids together is called a polypeptide. So a protein is usually made of several polypeptides wrapped together into a complex three-dimensional structure or shape. Once again, remember that the function of a protein is determined mostly by its shape or its structure. So that how those proteins fold up is critical to their function. We also talk about the directionality of amino acids or proteins. So because of the structure of an amino acid, a polypeptide chain has directionality. It has two chemically distinct ends. One of the ends in an amino group. The other end of the chain always ends in a carboxyl group. So that's true in all proteins. So we sometimes call this end the N terminus and the carboxyl end the C terminus. New amino acids are always added to this side, the carboxyl terminus of a growing polypeptide chain. So we always build from amino toward carboxyl. So another important thing to know for proteins are the levels of protein structure. We really break it up into 4 levels. Start with the primary structure, sometimes notated as 1'. This essentially refers to the amino acids in the chain and the order those amino acids are in. This is determined by DNA. DNA's only job is to determine the protein structure or the amino acid sequence of a protein. Yes. A mutation in the DNA can change the primary structure of a protein. And what that can end up doing, if the wrong amino acid gets put in the wrong place, that can completely change the shape and therefore the function of a protein. Let's move on to the secondary structure. This refers to the shape of specific sections of each polypeptide chain. Because amino acids are polar, typically, at least many of them are, they fold in certain ways. And hydrogen bonds form between different amino acids. This helps to hold the shape in place, and it helps to determine the function. So the polarity or non-polarity of the R groups is crucial in determining how a protein will fold. Proteins that are built on ribosomes that are located on the rough ER, that folding usually takes place inside the rough endoplasmic reticulum. The proteins that are made then are usually exported, and they either leave the cell completely or they end up in the cell membrane, embedded in the cell membrane. Other proteins are made by ribosomes that are just floating freely in the cytoplasm or the cytosol. These proteins end up staying in the cell and functioning within the cell that made them. The folding of these proteins usually takes place in these protein tunnel-like structures called chaperonins. And these are places where the proteins can fold without water interfering with that folding. There's two main types of secondary structure that we'll talk about in proteins, and those are called the alpha helix and the beta pleated sheet. Here we can see those. So all the dots that you see, these are hydrogen bonds between different amino acids. And they're helping to hold this polypeptide chain in this sort of helical structure. So this is an alpha helix. Here we can see the same thing down on the bottom picture. Again, hydrogen bonds all over the place. And these hold the protein in this three-dimensional pleated sheet arrangement. Again, remember, secondary structures are the shapes of just certain sections of a long polypeptide chain. Tertiary structure, sometimes abbreviated as 3', refers to the shape of one individual polypeptide chain, the whole chain, the shape of the whole chain. An important structure here, in addition to hydrogen bonds, are called disulfide bridges or disulfide bonds. These form between cysteine amino acids. So cysteine is an amino acid that has a sulfur atom in its R group. And if two of these cysteines get close together, these sulfurs bond. This bond is much stronger than a hydrogen bond, so it helps to stabilize a protein shape, make it much more stable, much likely to last and not be broken. Another thing that helps to determine the tertiary structure are ionic interactions between certain R groups. So there are certain R groups that are very, very, very polar, or not polar at all. But certain R groups are really positive or really negative, and this helps to determine how the proteins shape and fold. The last shape or structure of a level of protein structure is called quaternary or 4' structure. Remember, we said a minute ago that most proteins are made out of several polypeptides wrapped together. Here we see that on the bottom picture. So this is hemoglobin, the protein in your blood that helps to carry oxygen for you. And you can see that hemoglobin is made of 4 different polypeptide chains, all interconnected to form an overall 3-dimensional protein. That overall 3-dimensional shape of all the polypeptides wrapped together, those are called the quaternary structure. If any of those levels of structure change, that can change the overall shape and therefore the overall function of the protein, so it doesn't work the way it's supposed to. Denaturation is a common unraveling or unfolding of a protein. We oftentimes talk about this in terms of enzymes. When enzymes, which are proteins, denature, they don't work anymore. Denaturation can be caused by lots of things, but typically in living things, it's caused by changes in pH. And this can be either pH changes in the upward direction, the more basic direction, or pH changes in the acidic direction, the downward direction. It can also be caused by salt concentration or salinity changes, either up or down, away from optimum conditions. And the thing that we'll talk about the most in terms of denaturation is temperature. When proteins get hot, they oftentimes denature. Coldness is not going to do that to a protein, but increases in temperature above a certain optimum level is going to cause most proteins to denature. Denaturation almost always disrupts either the secondary or tertiary levels of protein structure by affecting either the hydrogen bonds, which are the weakest, or the disulfide bonds, which are the second weakest forces that hold together the shape of the protein. So denaturation is the unwinding or changing of shape of a protein. It's usually caused by a pH change, salinity changes, or increases in temperature, and it results in a protein not functioning the way it's supposed to. All right, the final thing we're going to talk about today are nucleic acids. So nucleic acids are the 4th group of macromolecules. They're also maybe the most varied in terms of their structure and their functions. Some nucleic acids are used to store genetic information. So here we're talking about DNA and RNA. Others are used to store and or transfer energy. And there we're going to talk about ATP, NADH, and NADPH. These two we'll talk a lot about during photosynthesis and cell respiration. And some nucleic acids like C, A, and P also function as cell signaling molecules. So very widely differing functions in the nucleic acids. Nucleic acids, most of them are polymers, and they're made of monomers called nucleotides. A nucleotide is essentially made of a 5-carbon sugar, which we can see right here, a nitrogenous base or nitrogen-containing base, which we can see here. And you've heard of these nitrogenous bases in regular biology. These are A for adenine, T for thymine, C for cytosine, G for guanine, and U for uracil. So those are the common bases we're going to talk about in nucleotides. And then on the other side is what we call the phosphate group, the PO4, usually a -3 phosphate group. So again, most nucleic acids are polymers. DNA and RNA are polymers. They're made of many of these guys, nucleotides, hooked together in chains or maybe multiple chains. And they're the primary sources of our genes and our hereditary information. DNA is made of a sugar called deoxyribose, 5-carbon sugar. We know it's made of 2 strands of nucleotides connected together. DNA in eukaryotic cells is always stored inside a nuclear membrane or nuclear envelope. And as I said a minute ago, its function is to code for proteins. It tells the cell, here are the proteins you need to build. Here are the order of the amino acids to build the protein out of. So DNA determines the primary structure of your body's proteins. Mutations end up with changes in protein structure, the order of amino acids, and therefore the malfunctioning of a protein in your body. You guys know that Watson and Crick were the guys who figured out the structure of DNA, and they figured out that it's made of 2 chains of nucleotides that are twisted together into a double helix. Those 2 chains are held together by hydrogen bonds between the nitrogenous bases. You guys probably know that an adenine and a thymine, those bond together. Those are a nitrogen-based pair. And a cytosine and a guanine, those bond together in a nitrogen-based pair. Again, those are held together by weak hydrogen bonds. It turns out A's and T's typically are held together by 2 hydrogen bonds. G's and C's are usually held together by 3. So it's a little harder to break apart a C and a G than it is an A and a T. They're a little more tightly held. What we call the backbone of the DNA molecule is made of alternating sugar and phosphate molecules. So here's a nucleotide. Here's another nucleotide. Here's a third nucleotide. These nucleotides are connected together along the backbone by a bond between sugar on this molecule, phosphate on this one. Phosphate on this molecule, sugar on this one. Phosphate on this one to sugar on this one. These bonds in the backbone are covalent bonds. They are very strong, very, very hard to break. Same thing on the other chain. Sugars and phosphates are connected. Now you will notice that the 2 chains are inverted. They run in opposite directions. On this side, the sugar molecules are pointing up. On this side, the sugar molecules are pointing down. And we're going to talk about how the 2 strands of DNA are anti-parallel when we get into DNA and the specifics of it. And that has a lot to do with how DNA is copied and transcribed. Remember, though, that the 2 chains are held together in the middle by hydrogen bonds that are weak. They're fairly easy to break. And you might think at first that you don't want your DNA being easy to break. But it turns out it's important, because for DNA to be copied, for it to be replicated, for replication to happen, the 2 chains have to separate. And it turns out also for DNA to be transcribed, or for transcription to take place, the chains have to be separated. Replication is when the DNA is copied. Transcription is when the DNA is used to make RNA, which then leaves the nucleus, goes out to the ribosomes, the protein-making factories of the body, and essentially functions as the directions for making your body's proteins. So let's talk a little bit about RNA for a minute. So RNA has ribose as its sugar, as its 5-carbon sugar. RNA molecules tend to be one-stranded, single-stranded. You'll talk about several types of mRNAs when we get into the genetics part of this course, of RNAs rather. There's messenger RNA, that's the one you're probably most familiar with. This is made from the DNA template during transcription. It leaves the nucleus, goes to the ribosomes, and functions as the directions for building proteins. Transfer RNA is another type of RNA called tRNA. Its job is to actually deliver the amino acids to the appropriate place during translation or protein synthesis. You'll also talk a little bit about ribosomal or rRNA. These actually build the ribosomes, the protein-making factories. They're what the protein-making factories are made of. So scientists can now sequence the nucleotide nitrogenous base sequences in genes of organisms and compare them. And what they can see is if they take the sequence from similar genes in different organisms, the more similar the two sequences are, the more evolutionarily related scientists would argue those organisms are. And you're going to talk a lot about this theme through the course. You're going to compare nucleotide sequences or amino acid sequences in related genes in different organisms. The more alike the sequences are, the more evolutionarily related the organisms are. You'll see that a lot, especially when you get to the evolution unit in this course. All right, the last nucleic acid we'll spend any time on today is ATP, adenosine triphosphate. ATP is not a polymer. It's made of a single monomer, a single nucleotide. It's made of a sugar, ribose, bonded to a nitrogenous base. In ATP, that's always adenine, always A. But then it's bonded also to 3 phosphate groups. And we can see the basic structure of ATP right here. The green would be the ribose. The orange is the adenine, the nitrogenous base. Here are 3 phosphate groups. ATP's role in the body is to store and transfer energy. You can think of ATP like your body's battery. It's what most of your body's chemical reactions run on. It's made during the process of cellular respiration. Think of cellular respiration like the process of charging up your body's batteries. And the energy to do that comes ultimately from food. As we said a minute ago, ATP functions to power almost every activity that occurs in the cell and in the body. And to do that, ATP is hydrolyzed. It's broken. When ATP is hydrolyzed, which we see down here on the bottom, ATP, one of the phosphate groups is removed and it's transferred to something else. That helps to transfer energy. What's left is ADP, adenosine diphosphate. So think of ADP like a dead battery. And exergonic reactions in your body, like cell respiration, charge that dead battery back up to ATP, which is your charged battery. So this is battery charging, and this is using your battery. And that cycles over and over again throughout your body. So ADP to ATP to ADP to ATP, that happens over and over and over. Charging, using, charging. Think of it as charging and then transferring energy. Charging, transferring the energy. Charging, transferring the energy. Over and over in your body. All right, we'll also mention this real quick. There are lots of cycles in the living world called biogeochemical cycles. You've heard of probably the nitrogen cycle, the carbon cycle, the water cycle. Today we're going to introduce the phosphorus cycle just very briefly. So phosphorus is really, really important in lots of living things. Phosphorus is a component of DNA in the phosphate groups, RNA, ATP, also in the phosphate groups. There's phosphorus and phospholipids, which make up your cell membranes. There's also a lot of phosphorus in bone. So phosphorus is really, really important. A problem with that is most of the phosphorus on Earth is found in rock. It's stored up in rock as phosphate groups. Well, the way that phosphate is made available to life is through weathering. So rocks will weather, water, wind, rain, eventually break down the rocks. Some of that phosphate or phosphorus is released into the soil. It moves through the soil with the water. Eventually, it makes it either into bodies of water or into the soil itself, and it's made available to plants or to phytoplankton in the water. These then use it to make organic molecules, things like phospholipids, DNA, RNA, ATP. So that the phytoplankton and the plants get their phosphorus directly from either the water or from the soil. And then when they die, decomposers break down their bodies, put phosphorus back into the soil. We as animals are going to get our phosphorus from eating plant-based materials or other animals that ate plant-based materials. Eventually, as the organisms die and are decomposed, some of that decomposed phosphorus remains in the soil. It may ultimately end up going back to rock, though. And you can see that happening up here in our phosphorus diagram. Point being, what you need to know about the phosphorus cycle, most phosphorus is stored in rock. It's made available to life through weathering, and then it cycles through living things from plants or algae to animals. All right, that's it for today's week 3 AP Bio content review session. I hope to see you guys again next week. Before we go, are there any questions I can answer for you?
Yes, what did you say about, go ahead.
Oh, I was going to say, is dishwashing detergent polar?
Let's see. So dishwashing detergent is used to break up fats. So let's see. Let me think about that for a second. It would be mostly non-polar, I believe, would be the correct answer there. What's the context of your question there? What are you trying to figure out?
Well, I usually use it to clean my glasses, and I use it's like a mild dishwashing detergent.
Right. So it's used to remove oils and stuff.
Right.
So that's meaning those oils are removed, and then it's used to break up the fats. It emulsifies them.
Mm-hmm.
So that's typically how soap works. It dissolves and then breaks the fats up into smaller pieces.
Okay. Thank you.
Mm-hmm. All right. I heard somebody else with a question.
I was asked about the last thing you said about the phosphorus. You said it was made of rock, and I forgot what you said after that.
Okay, not so much that it's made of rock. Most of Earth's phosphorus is found in rocks. That's where it's stored on Earth. So the way that it gets out and is made available to living things is that the rocks are broken down through weathering, through wind and rain and just typical environmental things. That puts some phosphorus either in the soil where plants can get to it or in the water where algae can get to it. And then once the plants or algae get it, they use the phosphorus to make things like phospholipids, DNA, RNA, ATP. And then we as animals get our phosphorus from either eating plants that got it that way or from eating animals that ate plants that got it that way. Does that make sense?
Okay, thank you. Yes, sir.
Other questions? All right, well, that'll do it for today. Hope to see you guys next week. Same time, same place, 10 o'clock on next Thursday.